This photograph shows a cluster of what we believe are neural cells derived from our adult cells, and we believe that this boxed region here represents one cell beginning to interact. Historically, most adult stem cells have been isolated from bone marrow, or some studies have shown adult stem cells isolated from the peripheral blood. We have taken this a step further and have isolated a very rare cell type that's found in the circulation, and we chose a rather unique approach to isolate that single cell. And furthermore, we believe that our approach has allowed us to isolate the cell in a very primordial state. It is a very early stem cell, and that gives it the potential to differentiate into a lot of different cell types. And unlike most other adult stem cells that have been described, these appear to turn into neurons. And so for a number of conditions, such as Parkinson's disease and spinal cord injury, these adult-derived stem cells may in fact be very beneficial because they do appear to go down the nerve pathway.
Oh, sorry.
Yeah, this is a really neat project because we were able to take adult-derived stem cells from the peripheral blood of pigs and be able to differentiate them into a wide variety of cell types. One of the most exciting, I think, is some of the neuronal cells that we've been able to develop. And another reason this is exciting is the line of pigs that we're working with has a gene in for the enhanced green fluorescent protein, which puts a mark on them so that we can then track these cells and confirm that yeah, these are really the cells we're working with and that they gave rise to the cells, in this case, in the rat brain.
The picture here on the left is a picture of one of our adult stem cells after we deliberately turned it into a neuron. In panel A here, this is a photograph of an adult stem cell. I believe that the phrase breakthrough is overused for a lot of research findings. I believe that this is another step in the stem cell discovery path. I believe that all discoveries stand on the shoulders of other discoveries, and this is yet another step in that long pathway. I believe that either our laboratory or others will build on this current publication, and I believe perhaps lead to the development of routine isolation of these primordial multipotent stem cells. Protein pig. We've injected these cells into a normal brain that's not fluorescent. So our next step clearly is to see if we can get this to work with humans. We have just started these experiments, and I am very optimistic that we will be able to achieve the same findings in humans. And when we are able to achieve that, then we can begin to really address whether or not these adult stem cells will be useful for a number of therapies. It is a very large concern of mine that our adult stem cell findings will be used by individuals who oppose embryonic stem cells. And I believe that that would be a mistake. Simply put, there are too many diseases that require too many different types of cells for therapy to put all of our eggs in the adult stem cell basket.